Illustrate the effects that diet can impart on the microbiota ecology, specifically the gastrointestinal system of humans and other mammals, and how can that have a further downstream effect in terms of host immunity and metabolism, specifically speaking about the onset of obesity or obesity itself. Now, allow me a few seconds to say why I was interested in this topic in the first place. I left my beautiful country, Peru, in order to pursue a biochemistry undergrad major in the United States because the topics of health and metabolism were always really enthralling to me. However, as I started to develop my studies as a biochemistry undergrad, I became really intrigued in this so-called second genome, the microbiome, which expresses the microbiota at different tissues in humans and other vertebrates, and what were its actual effects in health and metabolism as a whole. Now, allow me to elucidate what the microbiota is. It refers to the trillions of commensal organisms in different mucosal surfaces and tissues in the body. These tissues could be the skin, the mouth, the esophagus, the stomach, the colon, and the different reproductive organs in both male and female. Now, there's different phylums of bacteria that actually govern these different tissues, and that's important because these different microbial groups are actually going to exert different metabolic functions as well as play a role as immunologic modulators, which is a good way of actually introducing my second point. We actually have an alliance. We maintain a mutualistic relationship with these microbes in exchange for a place to live, which is what we provide and other mammals provide. They confer us with different proteins, mainly in the forms of enzymes, that allow us to digest and degrade different types of nutrients that we intake, as well as aid us in the biosynthesis of different macromolecules, which are essential for our survival. Now, the reason why these enzymes are important in the first place is because they are not encoded in the host genome, which is a primary evidence that evolution drifted us to coexist with these microorganisms. Now, further on, the microbiota is actually tightly interdependent with the host immunity. It is able to modulate the innate immune response, which is actually driven by phagocytic cells and antigen-presenting cells, which alert the body that's under attack, mainly by antigen presentation and the release of different cytokines. And it also is able to modulate either directly or indirectly what is the adaptive immune response, which are leukocytes, which are involved in clearing the pathogenic infection after 7 to 10 days of exposure. So based on this information, it wouldn't be unreal to think that the disruption of this host microbial interaction could actually bring repercussions to the host immunity and metabolism. And this now is where I introduce this new variable, dietary intake, not just food consumption in general, but any type of dietary intake. Is a leading factor that causes microbiota diversity, and the reason why this is because it affects us daily, right? We consume food every day, and some of us consume food and other supplements every day. And therefore, a bad diet is a predominant factor of the disruption of this important bacterial host interactions, as well as the disruption of the immunity and metabolism homeostasis in the host. Because of this concomitant relationship between the host immunity and the microbiota, there's a whole array of different syndromes that can arise in terms of immunity. However, in terms of metabolism, there is one prevalent illness that has affected the United States of America for the past 2 decades, and that is obesity, which usually counts by the hand of type 2 diabetes. Now, based on the information that I just provided, please allow me to introduce the 2 main goals for this review. I want to be able to amalgamate and put together the different factors that can arise of an aberrant immunity as well as the syndrome of obesity into one common denominator, which is the microbiota expression in the gastrointestinal system of mammals and humans as well. And also, and I think most importantly, I want to be able to address and illustrate how important is a balanced and healthy diet in modern society, especially in the USA society. Which brings me to my papers. Okay, so the first paper that I'm going to talk about was actually published by the University of Colorado in Boulder in 2005, and was actually one of the first articles that illustrated the effects of diet in the microbiota. This brilliant group of scientists back in 2005 used model mice, mus musculus, and decided to test more than 5,000 different ribosomal DNA consensus bacterial sequences in these rodents and blast them and compare them with already known human consensus sequences in the ceca or the intestine, of both mammals, in order to compare and contrast similarities between the humans and this type of rodents. And furthermore, they were actually able to knock out the leptin gene, which is a protein known to promote hunger inhibition, in order to be able to induce obesity easier in these rodents. And then compare this to the mice tested in this experiment were actually fed a polysaccharide-rich diet for a period of 8 weeks. As you can imagine, the knockout mice was fed a bigger proportion of this in order to increase their weights, their adipose tissue, and further on develop obesity. And the results are depicted above in the screen. As you can see, PCR analysis of different consensus sequences, as well as computational analysis of the different divisions of phylum bacteria in both mammals have revealed that actually humans and rodents share two phylums in common, Firmicutes and Bacteroides. And the reason why this is important is because both of them serve different metabolic functions. Firmicutes is actually involved in increasing the sensitivity for triglyceride synthesis, while on the other hand, Bacteroides is more involved in what is glycan metabolism. And so the scientists saw this and decided to do further experiments in terms of comparative analysis between the obese knockout mice and the slim type zeolins in terms of these two phylums of bacteria. And what they found out was that in terms of sequences, there was an upregulation in Firmicutes and an downregulation in Bacteroides. And it's impervious for the studies of obesity because it actually tells us that obesity is associated with an increase in sensitivity for triglyceride synthesis, as well as an aberrant glycan metabolism. Now, the second paper that I'm going to discuss was actually one of my favorites, done by researchers in Valencia, Spain, back in 2009. They took a more risky approach and decided to test the effects of a diet absent in gluten in 10 healthy subjects and the downstream effects that microbiota shift can do on innate immunity. They collected fecal samples before and after the intervention, the 30-day intervention, in order to do different sequence as well as immunologic analysis. Further on, they also decided to take into account the dietary composition of the volunteers before and after the gluten-free diet interventions because they wanted to know if there were any variables that could change in terms of energy balance and nutrient intake. And the results are here. Now, in terms of dietary composition, the only major change that they could see was that there was a decrease in polysaccharide consumption when the subjects were undergoing this different type of diet. Now, fluorescence in situ hybridization as well as quantitative PCR analysis yield an increase in general bacteria that belong to the phylum Proteobacteria. Now, Proteobacteria are actually involved in the biosynthesis of different vital vitamins such as B12, as well as enhancing the host metabolism as a whole. On the other side of the spectrum, there was a decrease in Firmicutes and Actinobacteria. Actinobacteria are also involved in biosynthesis, but of different intermediate metabolites in the host, such as anticoagulation factors. And so, just based on this microbiota composition, you can say, well, this result seems somewhat appealing until you look at what happens on the cytokine profile. Sandwich elastase is done on 4 enzymes. 2 of them promote inflammation. 1 of them a regulatory cytokine, and 1 of them a chemokine, CXCL8, which is secreted by epithelial tissue to recruit macrophages to the site of infection. All of them presented a substantial and significant decrease. And so what we can extrapolate from this article is that a diet that's absent in gluten and low in polysaccharide consumption gives parallel results with a reduce in innate immune response. Now, the third article that I'm gonna discuss by Dr. Hill and coworkers and friends in 2010 actually wanted to analyze the other side of the spectrum in terms of immunity. They wanted to know how detrimental were the effects of antibiotic in the gut microbiota of rodents, specifically colon microbiota, and what were the downstream effects in terms of innate immunity, specifically CD4 T cells. So what they did was they supplemented antibiotic with different strains of rodents for a period of 9 days. They collected their stools in day 10 for sequence analysis in terms of reverse transcriptase PCR and pyrrole sequencing, and also histological and immunological analysis in terms of flow cytometry. They also performed a quantitative analysis by Western blotting and quantitative PCR of this protein, RELM beta, which is known to be directly proportional to the levels of prokaryotic bacteria in the gut. And the results are here. In the top right corner, they analyzed different copies of ribosomal DNA by reverse transcriptase PCR. And what they found out was that in the experimental group, the rodents that were fed the antibiotic, there was a almost tenfold change between day 9 and then day 9 and then 1 in bacterial ribosomal copies. Now, in terms of quantitative analysis and Western blotting, this protein, RELM beta, and therefore prokaryotic bacteria, presented a sudden decrease as time started to dilate, and of course, as more antibiotic was introduced into the rodent systems. Now, figure A to D represent the relative expression of mRNA, the different expression and release of the cytokines that I'm about to talk about in the mesenteric lymph nodes and the intestinal lamina propria, and a statistical analysis in terms of percentages in the column of 2 cytokine profiles. One of them, interferon gamma, which is secreted by the helper cells type 1, which are CD4 T cells that have seen their antigen, have actually proliferated, and they're able now to deal with intracellular infections with their antigen presentation and infection. And moreover, also these other cytokine, interleukin-17A, which is secreted by T helper 17 cells, which are also CD4 cells, CD4 T cells that have seen its antigen, and are now able to generate a better innate immune response. However, they're part of the adaptive immune response. And as you can see in all figures A to D, there was a severe and substantial decrease in the release of these cytokines, especially in terms of expression of RNA. So there's 2 home messages I actually took from this article. The first one is that a depletion of bacteria in rodent gut microbiota is actually associated with an aberrant adaptive immune response. And moreover, an excessive amount of antibiotic intake is actually increases the pathogenicity, as well as dilates the time that the microbe is able to infect the host because the adaptive immunity is not working so well. Now, the last paper that I'm going to talk about by Park and friends in 2013, they actually wanted to offer a new insight in terms of this type of experiment. They wanted to see if genetics was able to exert certain control in the microbiota plasticity. And moreover, if an external stimulus such as diet was introduced, they wanted to know if genetics was also able to control the onset of obesity. Moreover, they also tested which factors, both environmental and genetic, were able to be related of obesity, and if there were any obesity traits that overlapped between humans and rodents. And the way they did this was they used 100 different types of inbred mice strains, and they fed them a high sucrose, high fat diet for a period of 8 weeks. And they also performed further genomic-wide association studies, as well as other global gene expression studies. And these were their results. They found out that in terms of this particular obesity trait, which is body fat percentage growth, they saw that there was a difference between 0 and 400% within the inbred mice strains. And so they concluded that genetics in these rodents are able to control of about 80% of this trait. Moreover, they found that there were 2 chromosomes, chromosome 2 and chromosome 6, there were factors for obesity. Specifically, chromosome 2 was loci. They found 2 loci, in 2 chromosomes, chromosome 2 and chromosome 6. The first loci in chromosome 2 was actually associated with the synthesis of phospholipids. And chromosome 6 loci contained 11 genes. All of them were factors for controlling the gene expression in adipose tissue. And so, based on these results, the main take-home message that I could think about was that genetics is also able to exert certain influence on the onset of obesity. And so, well, why should we care, right? Why does this matter in the first place? For the scientific world, why it matters in terms of obesity should be extremely straightforward by now, right? It's an epidemic in the United States of America. The American National Health Society, back in 2012, extrapolated data. 2012, that's 4 years ago. Extrapolated data that 1/3 of adult Americans were actually obese, and 16% of that 1/3 were candidates for morbid obesity. Morbid obesity is characterized by a body mass index of over 40%. And moreover, this association also extrapolated data that in a period of 10 years between 2012 and between 2002 and 2012, the numbers of this type of obesity had actually doubled from 7 million to 14 million. Moreover, this syndrome also affects children and, of course, also affects children and adolescents as well. Ranges from 2 to 19 years of age. The rates have also increased in this same time period from 2002 to 2012, from 15% to 20% of all children and teenagers. So there's a lot of reasons why we should be concerned about this. Or you guys should be concerned about this. So why does it matter? My country's great. Why does it matter in terms of immunity? Well, as I mentioned before, there's different species and different types of phylas of bacteria that govern different types of mucosal tissues. And guess what? The pathogens love to infect humans through these mucosal tissues. Now, I only described and talked about the gastrointestinal system in humans and rodents, which are both mammals. However, as you can see from the worldwide deaths annually from mucosal infections, pathogens also use the respiratory tract and the urinary tract to infect different types of mammals. So it's just something to keep in mind of. And finally, I decided to put my approach because this is my presentation. First of all, I thought, why don't we actually be able to account ourselves for a diet in which matches our energy balance and nutrient intake? Why don't we just do this to avoid all of these problems? Well, turns out it is somewhat difficult now, because 50% of all the processed products in the supermarket actually contain high fructose corn syrup. But that's not even the biggest problem. The biggest problem is that a lot of people actually have a misconception of what health is. I've been living here for 4 years, okay? And then Colorado is also considered the slimmest state in the country. However, I ask parents all the time about what they think is the most beneficial things for the kids in terms of the health, and a lot of them conflate their child being overweight or obese with being healthy, which is completely inaccurate. But again, I can't blame them for this, because we actually enhance this type of behavior. Look at this, heart attack grill. If you weigh over 250 pounds, you eat for free, which can be hilarious in other types of scenarios, but not here, and not in the science world. Because all this amazing effort that all of the scientists have provided us for several years in order to increase our knowledge of what the microbiota is worth nothing. It's worthless if we don't actually are able to change the people's conception about what health is, and if they are not able to change their behavior. I want to thank Dr. Khaled for being extremely patient with me and for allowing me to camp inside his office in several occasions. I want to thank the faculty who came here and my friends. Hey guys, sorry, I lied, there's no free cookies. But thanks for coming. And I want to take time now and actually thank my parents for this amazing 4 years of both financially and emotionally support. I don't know if someone's taping me, but I want to thank my mom for being the greatest inspiration of my life. And I really want to thank my dad for always having the enormous courage to show me and to pull me away from the horrors of mediocrity. Muchas gracias indeed. Thank you guys so much. It's been a wonderful 4 years. Yes, Tyler.
In regards to, I think it was your third paper with how antibiotics influence your microbiota.
Yeah.
Did you read anything that, that slide. Did you read anything that would indicate how long it would take for your gut microbiota to get back to where it was, where you get Th1 levels back to normal? Like about how long after you get off antibiotics do your Th1 levels get back to normal?
Well, again,